Hey guys. I'm having some computer difficulties here. Can you guys hear me? Yeah, okay. Yeah, I'm having some computer difficulties here, so give me a minute. Computer is moving at a glacial pace. I want to think it's my computer, but it might also be just Moodle is dragging this morning for some reason. I just posted a new set of amphibian reproduction slides, so you can go and access that. I'm not sure how much we're going to get through today, because we also have some bookkeeping things to work on. Okay. All right. First thing, when you look at the Moodle site, let me get back to the Moodle site. You will see up at the top of the Moodle site the herpetology schedule. If you've been paying attention to it, which Kelsey has, thank God Kelsey has been paying attention to things, and I'm serious about that. We're behind. Not surprising. I was out for a day, and we were already about a half a day behind anyway at that point. So I put in a new schedule, basically just rearranged kind of what was left of the things that we still have to cover. So what I've done is, let me share screen. So what I've done is I've just put over here in column E is kind of the plan for the rest of the semester. So we had an exam scheduled for Friday. In the past, exam 3 has kind of covered up through reproduction, and I kind of like to keep things sort of thematically organized if at all possible, which is why we didn't add new material onto the tail end of exam 1, for example. So I would like to finish reproduction in amphibians and reptiles, and I think it will take us through Monday in order to get that done. So if it's okay with you, and I need to see your faces now in order to do this, I would like to put off, and I don't do this very often, I would like to put off exam 3 until the 16th of April. Do you guys have any difficulties with that? Okay, good. All right, so let's do that. The other thing that we'll do is there was a literature summary scheduled for Synervo that was never posted. It was a paper, it is a paper that talks about reproduction in a lizard species out in the desert Southwest. He did this cool thing when he was a graduate student where he actually manipulated the size of offspring by basically going into the females and puncturing their developing follicles so that they would yolk up fewer eggs. And as a result of yolking up fewer eggs, they would put more energy into those eggs. Those eggs would get bigger and they would produce these gigantic offspring. And then he would take eggs after they were laid and he would draw off yolk. So he would take yolk from the eggs, and that created these little tiny lizards. And so he expanded the phenotypic range of what baby lizards are, from little tiny lizards to normal sized lizards to gigantic lizards. And then he let them loose on the landscape and kind of looked to see how they survived. It's an interesting paper, but in the grand scheme of things, since we're running behind, you probably, well, I don't know. Will you complain if I don't have you read Cinervo et al. 1993 and don't have you write a summary over it? Okay, we're getting agreement on those things. So let's just scrap that. It's interesting paper if you're ever interested in kind of, his question was basically, how do you know that an organism has kind of arrived at the evolutionarily optimal body size for offspring in terms of their investment decisions and things like this, if all you have is natural variation to work with? Maybe lizards would be better off if they were much bigger than they actually are when they're hatched. Maybe they're way worse off if they're smaller, and that might explain why lizards invest in reproduction the way they invest in reproduction. But the only way to do that is to observe lizards that are born outside the normal range of variation, and that's what he was attempting to do. Pretty cool paper, but we can probably live without it in the interest of time. So those two changes. Third change, tomorrow you were supposed to have a annotated bibliography to me, but I haven't yet ground through your, sorry, not annotated bibliography, a dichotomous key. I have not yet ground through your previous dichotomous key. Kelsey's like, what annotated bibliography? Sorry, I've got too many.
I really thought I had missed something. I was like, oh dear God.
No, I misspoke. Dichotomous key. So you're supposed to have a dichotomous key tomorrow for the reptiles. I'm still grinding through the dichotomous keys from the amphibians because it takes a while to work through them. They're long for one thing, and they're complicated, and I have to think a lot about them because they're also different. So I would like to put off the dichotomous key being due so that you have feedback on your current dichotomous key for amphibians. I would like to put that off until the the 22nd of April. That's a Thursday. We don't have lab that day, but that's a Thursday day. My goal would be to get your amphibian dichotomous keys back to you beginning of next week, and that'll give you time to take whatever feedback you get on those and alter your reptile keys. Does that work for you guys? Okay, then I'm going to make another change out here. A new due date for that. And after class, I will upload an updated version of this. So remember that on the 22nd of April, that's a Thursday, we will be having class as if it was a Friday, because on the 23rd, that is undergraduate colloquium day. 2 of you are presenting at colloquium, is that the case? Kelsey and Elizabeth Ellis? Yep, they will be presenting. Kelsey, you're presenting about something having to do with Spanish, right?
Yeah, my research essay from last semester for Dr. Wells.
Okay, and Elizabeth, you're presenting, what's that?
Spanish is my other major. I'm a double major.
Yep, I knew that. Elizabeth, you're presenting on your senior thesis work, is that correct?
Yeah.
Okay. All right. So on undergraduate colloquium day, you at least have 2 colleagues from the class who are presenting things that you can go and see. Go and see. I mean, by that I mean sit at your computers and watch them present. So that's going on that Friday. But we'll just make the dichotomous key do that day as if it was a lab day, even though it's not. Tomorrow in lab, we will be going into the field. We will be carrying camera gear with us. We will learn how to take photos of amphibians and reptiles, although probably most of what we will see tomorrow is in the reptilian realm. Then on the 29th, we'll get another week of field work, and then that'll pretty much be the end of lab after that. I'll also, let's see. Oh yeah, we'll have the 15th. So this week and next week we'll be going out into the field, and then the week after the colloquium day week, we'll go out into the field. And over the course of those 3 weeks, you should be able to get all of your collection done. On the 15th, we will go to a site that has a lot of amphibians, and it's an amphibian that has tons of the most common amphibians that are in this part of the country. And so I don't have any problems with you collecting live specimens to practice the process of collecting. So tomorrow we'll do all photography. The following week we will do all collecting, and then we'll come back into the lab early so that I can show you how to preserve specimens and pose them and get them ready to put in a jar and put into a collection. And then the 29th, we'll go out to another site that's over here near Missouri City, and we'll do some collecting there as well, but that's probably going to be mostly camera work. And tomorrow I do have a van. So I checked out a van. We will be driving to Blue River Glades, which is down in Swope Park. If you have concerns about hopping into a van with a bunch of your colleagues, given that we're still under COVID times, feel free to drive your own car if you would like. We will just spend a couple of minutes up in lab, and then we'll go out. The van will be parked in the parking lot here. And so if you're down the hill, you can run, get your car, and just follow us to Blue River Glades. If you have any other concerns about being in a van, then by all means, feel free to drive your own car. All I care is that you're at Blue River Glades when I'm at Blue River Glades, because you probably don't know where Blue River Glades is, and I will show you around. So either in the van or in your own car, we will be going to Blue River Glade tomorrow at 10:45. I will keep track of the amount of time that it takes to get down there so that we return in time for you to get to your next class. So that will be that. Given the weather today and the forecasted weather tomorrow, I would come prepared to get wet. And at the very least, I would come prepared to walk through mud in some cases. So, you know, do not wear your fancy shoes. I will probably be there with jeans with holes in them and a ratty shirt and my hiking boots. So if you have a pair of gloves, you might want to bring a pair of gloves for flipping rocks and things like that. If not, don't worry about it. It's not that big of a deal. But come prepared to spend the whole time period out in the field. Let's see. What else do I need to take care of logistically? Can you guys think of anything? Okay. All right. Last time, which feels like forever ago now, we were talking about amphibian reproduction. So let me go and pull up my slides. So I don't actually recall exactly where we were, but we had gotten started into talking about different modes of reproduction. And I wanted you guys to think about reproduction in amphibians within the context of this idea of constraint. In amphibians, they have unshelled eggs. So the eggs are not protected by either a parchment shell or a calcareous shell. And as a result, they're prone to desiccation. And so as a result of the structure of the eggshell and the fact that they didn't go through the process of evolutionarily developing an amnion, which protects the embryo from desiccation, you generally find that amphibians are more closely tied to water. And so this might influence all of these kinds of things. And we talked about these last time we met. It might influence the mode of fertilization, the mode of reproduction, whether you're oviparous or viviparous, the mode of larval development, whether you're free living larval stages in a pond somewhere or whether you're going through direct development. It might influence parental care. Do you have no parental care? Do you guard your eggs? Do you beard your eggs? Or you can also think of viviparity as a form of parental care. And then the specifics of egg laying. Where do you lay your eggs? Do you lay them in water? Do you lay them in foam nests? Do you lay them adhered to leaves? Do you lay them just sitting out on land, et cetera, et cetera? So these are all things that are found in amphibians, but these things are not necessarily independent of one another, and these things are influenced by the environments that these organisms live in. And so we had begun the process of going through and thinking about fertilization. And if I recall correctly, we were working through internal versus external fertilization, weren't we? That's where we finished. We were talking about internal fertilization in frogs. Yeah, okay. So we know about internal fertilization in salamanders. They lay the males put down spermatophores, this gelatinous glob with a ball of sperm on top of it. The female comes along and will position her cloaca over the spermatophore, draw the spermatophore up into her cloaca. She will store the sperm in the spermatheca and then use the sperm that are stored in the spermatheca to fertilize the eggs as they pass out of her. So fertilization in salamanders occurs internally, but in most cases it occurs internally, but just right before the eggs are laid. This has lots of implications for male behavior in that males don't necessarily have much control over paternity. And so one of the things that drives male decision-making with reproduction is paternity assurance. If you are a male salamander and you're mating with a female salamander, she may have already mated with multiple male salamanders before you and stored their sperm in her spermatheca, and she may mate with multiple male salamanders after you and store it in her spermatheca. And I remember I told you last time that they can actually pick and choose whose sperm actually are used to fertilize these eggs in some cases. It's something that we refer to as cryptic mate choice. This is different, though, than in external fertilization, where the eggs are fertilized after they are released from the female, in which case the male has a high assurance of paternity. The female just laid those eggs, and I sprayed my sperm over them as they were exiting her. As a result, I know that those eggs have been fertilized by me, as opposed to in an internal fertilization situation where the eggs that she's producing might or might not be fertilized by me. In which case, you might see things like male guarding, mate guarding, where males guard their mates until they go through the process of laying eggs, once again in an effort to prevent that female from mating with males after that male has mated with her. So there are all sorts of behavioral things that happen in all of this. So external fertilization is the exception to the rule in salamanders, and I mentioned that it only occurs in these two families, and these two families are monophyletic families, and they're different from all of the other families in a phylogenetic sense. These are among the earliest salamanders to branch off from the rest of the salamander group, and the Hydnobiids, which is a Old World group, and the Cryptobranchids, which has representatives in the Old World and the New World. These two families are the only two families that have external fertilization. We briefly mentioned internal fertilization in Caecilians, the presence of the phallidium. It's an intermittent organ, and so this is a different form of internal fertilization, though, as opposed to the salamanders, where there's the laying down of the spermatophore. This is much more kind of the analogous to a mammalian penis, essentially, where the male inserts the phallidium into the female and transfers sperm that way. The phallidium has all kinds of different shapes. They can be used in phylogenetic analysis to tell different species apart, much in the way the hemipenes of snakes and lizards can be used. We'll talk about that when we get to reptile reproduction. So we were then moving on. I think this is where we stopped last time. There is internal fertilization in Anurans, but once again, this is the exception rather than the rule. I think your book tells you that there are 3 species that experience internal fertilization, and they concentrate primarily on 2 of these. One of these is the tailed frog that occurs in the Pacific Northwest. What is the tail made up of? What's the morphological basis of the tail in tailed frogs? It's an outgrowth of what structure?
Isn't it the cloaca?
Yeah, it's an outgrowth of the cloaca, and the male has it and the female does not. And the male uses it as an intermittent organ, so he will insert his tail in this case, into the female and transfer sperm that way. This is a Leptodactylus coqui. This is a tree frog in the family Leptodactylidae, which occurs in Puerto Rico. It also goes through internal fertilization, but it doesn't have an intermittent organ. It doesn't have any sort of copulatory organ. The female in this case will wrap her legs up around the backside of the male. The male will position his cloaca opposite the female's cloaca and transfer sperm from cloaca to cloaca. This is kind of similar to the way that birds mate. If you know anything about bird morphology, they don't have a penis or any other type of intermittent organ, with the exception of ducks, if I recall correctly. It's been a while since I've taken ornithology. But they have cloacas as well, and they do a thing that ornithologists, I guess, cutely refer to as a cloacal kiss, where they basically press their 2 cloacae up against one another and transfer sperm that way. And so this odd copulatory behavior, this odd amplexus behavior, where the female is actually grasping the male, pulling his cloaca towards her cloaca and transferring sperm. So it does occur in Anurans, but it's really rare relative to all the other species that are primarily external fertilization. So we talked about amplexus a long time ago when we were talking about the diversity of amphibians. These are the 3 forms of amplexus in frogs and toads, and they're all based on where the male is grasping the female. So in amplexus, it's always the male on the back of the female, and the question is just whether the male is grasping the female around the base of the back legs, in which case that's inguinal amplexus. Are you grasping the female behind the front legs, in which case it's axillary, or are you grasping her around the head, which is cephalic? So in this case, you can see the male's arms in front of the front pair of legs of the female. In all of these cases, what it serves to do is it serves to position the cloaca of the male above the cloaca of the female. And as the female expels her eggs into the water or into a foam nest or anything else, the male will then squirt his sperm over the eggs as they exit the female. What's another thing that you notice about these photos of frogs and toads in M. plexus?
The male's smaller than the female.
Yeah, so in at least two of this, well, and really in all three of these cases, the males in this case happen to be smaller than the females. In this case, this male is much smaller than this female. These are the horned frogs, and is a member of the genus Ceratophrys from South America. So there's the sexual dimorphism in size. Thinking evolutionarily, why might there be a consistent sexual dimorphism in frogs and toads, and the sexual dimorphism is such that the females are almost always larger than the males? What might be a good reason for that? Think about gamete production. What are the gametes of males like relative to the gametes of females?
Male gametes are smaller.
Male gametes are much smaller. So sperm are tiny relative to eggs. In my animal behavior class, when we talk about reproductive patterns and kind of the overarching sort of thing that drives male and female reproduction is, if you think of an egg and you think of a sperm, in terms of investment, females, even before anything else happens, just in the process of producing an egg, females have already invested way more energy in eggs than males have invested in each individual sperm. As a result, males tend to produce tons of low energy, low nutrient requiring sperm. Females tend to produce a fewer number of higher energy, higher nutrient requiring eggs. And then this influences all kinds of behavioral, there are all kinds of behavioral implications of that initial investment difference. How does this then result in sexual dimorphism in frogs and toads, where in general, the male frog is smaller than the female? These are frog egg masses. What's the advantage of being big and female? Big if you're female.
Do you produce more offspring?
So the bigger you are, the more eggs you can produce in theory, and of the eggs that you produce, you could potentially make them bigger. And so you may be able to produce more eggs, or you may be able to produce bigger eggs, or you may be able to produce more and bigger eggs. And if you're a male and you're producing these little tiny sperm, being bigger in size doesn't necessarily gain you a lot in terms of producing more sperm, simply because all of your sperm are little tiny things that don't require much energy at all. And so you might have natural selection selecting for larger females because it allows them to put out more offspring, or larger offspring, or both. And so you'll oftentimes see, well, it's the general rule, male frogs are considerably smaller than female frogs. What are conditions where you might see an exception to this rule? And this exception doesn't occur very often in frogs, but it will occur in lizards and snakes to a certain degree. When is it advantageous to be a large male with reference to reproduction?
Like if they're competing for territory or for a mate.
Okay, so anytime you have male-male competition, either for territories or for mates, you might expect males to be larger than females because selection, once again, is driving larger male size because the bigger you are, generally, the better you're going to be able to hold on to territories, and the better you are going to be able to compete for mates. And so, for example, in a lot of the plethodon and salamanders, we don't see sexual dimorphism in body size at all. And that's because in plethodon and salamanders, in many cases, the males are maintaining territories. They're competing with other males for access to females in their area. And as a result, the sexual size dimorphism isn't present because males are involved in male-male competition for mating opportunities. So this just gets us into thinking about behaviorally what's going on with males and females. So when we think of modes of reproduction, we can think about the type of egg that is being produced and how the embryo is being provisioned. We can think about whether organisms are oviparous versus viviparous, and we can think about where organisms are laying their eggs. And so your book mentions that you can think of two different egg types. One egg type is involved in what is called lesotrophy. Lesotrophy is the mechanism of provisioning the offspring such that most or all of the nutrients and energy that the offspring will need for development until hatching are provided by, or until metamorphosis are provided by the egg. Matotrophy implies that the feeding of the larva is going to be done by the female. And so oftentimes what you see with lesotrophy is the eggs are bigger, they have higher levels of yolk. In matotrophy, the eggs are smaller because the larvae are going to hatch out at an earlier developmental stage and then feed on, for example, the lining of the oviduct or secretions of the oviduct or something like that. So the type of egg has to do with the way the embryo is being provisioned. Obviously, if you're involved in matotrophy, once again, this is one of those lack of independence things. If you're involved in matotrophy, you're more likely going to be viviparous because the larvae hatch out inside of you and feed on the secretions of your oviduct, as opposed to lesotrophic species will tend to have oviparity, where you're laying eggs and you're not provisioning the embryo from your own body beyond the yolking of the eggs. And then when we think about egg laying sites, there are a variety of places where amphibians can lay their eggs. And this is actually where we see probably the greatest degree of diversity among groups. So before we get to that, your book has this weird diversion as it's talking about these other things, and it begins to talk about methods of fertilization. And so it brings up two cases. One is a hybrid frog complex that occurs in Europe. So these two ideas of hybridogenesis and kleptogenesis, and they've changed the genus of this frog species And I don't remember what the new genus is, so let me look it up real quick. Oh yeah, it used to be in the genus Rana. So Pelophylax esculentus. This is it's a pond frog. Clearly, it's a species in Europe that has undergone formation of a new species due to hybridization between two parental species. So, like we've talked about with the Teid lizards, you can have new species arriving through hybrid fertilization of two parental species. Oftentimes, this comes with changes in ploidy. And so you get mix-ups in meiosis such that oftentimes you produce gametes with diploid genomes in the gametes, and as a result, you might end up with, for example, tetraploid hybrid offspring. This is also what has happened with the gray tree frogs here in the United States. Those are two species, one of which is presumably a hybrid offspring species of the other species. And so hybridogenesis is one variation of fertilization mode that has resulted in the formation of this particular species. What is crazier and weirder, though, is something that occurs here in North America. And in North America, we have this group of salamanders that occur up in the Great Lakes region. And this is a form of what your book refers to as kleptogenesis. And what happens is that there are a set of unisexual ambystoma groups that are all sister taxa of dioecious groups, male and female groups. And these unisexual species actually breed with the dioecious groups. Dioecious just refers to two houses. It's a term that we usually use in reference to plants. When we think about plants, you can think about a plant as having flowers that have both male and female parts. In some cases, you can have flowers where there's a male flower and a female flower, but those male and female flowers live on the same plant. And then there are some species like ginkgo trees where there is a plant that produces nothing but male flowers, and then there's another plant that produces nothing but female flowers. And whenever we have those male and female flowers in two different individuals, we refer to those as dioecious. This is how you spell the word. It's a weird word. The dioecious species are those where the male and female flowers are on different individuals. And so you can also think of animals as being dioecious or monoecious as well. And in this case, we have some species like Ambystoma tigrinum, where there are male Ambystoma tigrinum, there are female Ambystoma tigrinum, and then we have a set of Ambystoma that are unisexual. They are all female. Now, these all females species will mate with male species, with male individuals of the other species, and this is what prompts them to reproduce. But in all of these cases, even though they may absorb the male genome, ultimately in the process of forming new offspring, those male genomes are rejected during fertilization. And so it's a really pretty weird situation in that copulation goes on, but the genome isn't actually being utilized because these things are reproducing parthenogenically. And so if you look at the mitochondrial DNA, which once again, mitochondrial DNA is just passed down from female parent to female offspring. You don't get your mitochondria from your father, you got them from your mother. And you can basically trace the relationships among these different groups, and all of the unisexual ambystoma form their own monophyletic clade here, and they're separate from all the rest of the bisexual, the dioecious species of ambystoma. And this is called kleptogenesis because when the female unisexual organisms breed with the males of the dioecious species, they basically are taking their genome from them, but that genome actually doesn't get passed on to any offspring, and so it's a form of stealing the genome from another species. So when we think about egg laying, we can think about where eggs get laid. And so in salamanders, we can have aquatic eggs, and they can either be laid in still water or in flowing water. This is what your book refers to as mode 2 and mode 1, mode 2, and then mode 3 are individuals that lay eggs not in water. And so when you look at aquatic eggs, they look different. And so here is an egg mass from a salamander that is laying eggs in a pond or in an ephemeral pool or something like this. You see there's just a big glob of eggs encased in this gelatinous material, attached in this case to a stick, but they're basically large masses, large amorphous sorts of masses. If you find species that lay eggs in swift flowing streams, though, you will find that generally they're laying fewer eggs, and the eggs will be encased in this kind of streamlined packet rather than this big globular packet. And they're attached to the undersides of rocks or the undersides of logs and things like this. And presumably this streamlining of the egg mass prevents it from being washed downstream in swift flowing water. So salamander eggs laid in water, still water eggs will look different than eggs that are laid in rapidly moving water. And so that's the reason for these different designations, mode 1 versus mode 2. And then among mode 3, you have lots of different ways in which things happen. So you can have terrestrial nests, and the terrestrial nest results in larvae that feed in ponds, or larvae that get moved to water, or non-feeding larvae, such that you're going to have direct development of individuals in the plethodonids. Arboreal nest species, once again, these are all plethodonids, where there is a nest up in a tree or a bush. The eggs are oftentimes adhered to the leaf through sticky coatings of the eggs. This, once again, involves direct development and is only found in the plethodonidae. And then you can also have non-aquatic eggs that are non-aquatic because they're just retained in the oviducts. And then you have viviparity, and this is in members of the Salamandridae. So these are examples of some eggs laid outside of water. I've mentioned Ambystoma opacum. This is Ambystoma opacum, the marbled salamander, one of my favorite salamanders. These occur in extreme southern Missouri, but none of them get up here. What happens with Ambystoma opacum is the females move to areas where there is a small depression, where later in the winter, late winter, early spring rains will fill up that depression. But back in the fall, which is when they lay their eggs, these depressions are dry. And what the female will do is she will lay her eggs underneath a log, and then she will guard them throughout the fall and over the winter. And then when the spring rains come and fill up that depression of water, the eggs will hatch at that point. They've been undergoing larval development inside the egg during this time. The larva will hatch, and then she will leave the nest and go crawl back into the forest. In Nacogdoches, Texas, where I was an undergraduate student, there was a pond an ephemeral pond in a experimental forest near the campus, where you could find a lot of these animals in the fall sitting on their eggs. This is a species of Bolitoglossa. It's a plethodonid salamander from the New World tropics. It has laid its eggs on a leaf, and once again, in this case, the male is going through guarding behavior of the eggs. Once again, these will be direct developing eggs. They'll develop into miniature little juvenile salamanders when they hatch. But once again, this is reproductive mode 3 from this table, non-aquatic eggs. In one case, Ambystoma opacum, where the larvae will feed in ponds, and in the other case with Bolitoglossa, where you have direct development in an arboreal nest. And then these are examples of frog nests, and so that's the next thing that we need to talk about. These are typical Ranid style frog masses, where each of these masses is laid by an individual female, and it just so happens that there were a lot of females in this particular site laying eggs. This is a close-up of what an egg mass will look like, and then this is a peculiar thing that happens in frogs, referring to foam nests, which we'll talk about. So you can think about Anurans in a similar sort of way, as you can think about them in salamanders. You can have eggs that are laid in aquatic environments. You can have still water or flowing water, and the tadpoles are what we call the larval form of frogs and toads. They'll either feed in the ponds or feed in the streams. But there are lots of variations. You can lay your eggs in a subaquatic chamber. You can lay your eggs in a basin that is near water, and we'll see an example of that. As organisms made this transition from breeding in the water to breeding on land, there were these intermediate situations where you might breed in really small bodies of water that were adjacent to larger bodies of water. You can breed in bromeliads or tree holes, these plant structures above ground that hold water. Lots of different variation. You don't need to know all of these. You just need to know that there is a lot of different ways of going about doing these things. A lot of the Leptodactylid frog species will create foam nests. These foam nests can either be floating on the surface of water, or you can also have foam nests that are attached to plant material, attached to leaves and things like this. And then there are some species that actually lay their eggs in the body of the female, on the surface of the female's body. Their back will grow thick layers of epidermis that will basically grow up and encase the eggs, and this occurs in the family Pipidae, and we'll see an example of that. Actually, let's just go and look at that. This is an example of one of the frog species that lays its eggs in a small depression of water, but this is adjacent to a larger pond, and so this will dry out relatively soon. But what generally happens is the animal lays the eggs in this small depression of water. They're protected in that small depression of water because it tends to dry out, so there aren't any predators in that water. When the larvae hatch out about the time that rains are coming. The rains fill up the adjacent pond. That adjacent pond takes over this small depression of water, and then those larvae are released to the larger pond after they're old enough to kind of fend for themselves. You can have organisms breeding in cavities underground, which once again, the eggs are underground. They're not necessarily in an aquatic environment down in there, but they might be if the water table is high enough. But what will happen is over time, as rains come, this will fill up. It'll be covered in water. That water will fill this cavity, and the tadpoles will emerge from the hole. In this case, you have this gelatinous mass that is attached to the underside of a leaf, and as the tadpoles hatch, and you can see them developing here, as the tadpoles hatch, they will break down this gelatinous mass and they will drip out of the bottom of this, and they will drip into a water body that is below the leaf, up a pond or a stream or something like this. I was hoping to show you some foam nests. Here's a foam nest. So here's a foam nest. This foam nest is formed by the male and female kicking their legs as the female expels her eggs, and as the male expels his sperm. They basically whip all of that stuff into a froth, and the eggs are encased in that. The eggs are encased in that. The froth itself basically starts to crust up on the outside and provides this protective layer in which the eggs develop. As the eggs hatch out, the larvae hatch out, they will then begin to break down the gelatinous structure inside of the nest until the nest finally breaks down to the point that it releases the larvae. In this case, this is one that looks like it's on the forest floor, but you can also find these once again in plant material above a body of water, and you'll see the larvae just dribble out of the foam nest. So we have a variety of ways of having aquatic eggs. We have a variety of ways of having non-aquatic eggs. Anybody I want to call attention to? No, but I want to go back. In this situation, there's this example, Reproduction. This is a species that lays eggs in flowing water, and one of the ways that it copes with keeping those eggs safe in swift flowing water is that this is what we refer to as this is called the midwife toad, actually. And she actually will swallow the eggs, and the eggs incubate inside the mother's stomach. And then when they give birth, they actually have the froglets coming out of the mouth of the female because they've been developing in her stomach. So in the case of non-aquatic eggs, not surprising that there are some species that exhibit direct development. Once again, this is a arboreal nest. More non-aquatic eggs are found in foam nests. So we have all kinds of foam nests. Once again, you will oftentimes see that these are in the family Leptodactylidae. That's a pretty common thing within that family. We have eggs laid on the outside of males. We have eggs laid on the outside of females. And then we have eggs that are retained in the oviducts of different species. So once again, if it could be thought of, it's probably been done somewhere in the amphibians. There's quite a lot of parental care, especially in these frog species that lay their eggs away from water. Anytime you're laying your eggs on land, you either have to have a way of getting your offspring to water after they hatch, or the easiest thing is just to have them develop into little miniature versions of themselves, and then they can hop to wherever they want to after they have gone through direct development. Now, if you are a frog that lays its eggs on land and free-living larvae hatch out, one of the things you have to do is you have to figure out a way of getting those larvae to water. You can do that by laying those eggs above water such they just dribble out of the nest into the water. But what's a way in which dendrobatid frogs, these are the poison dart frogs, how do they accomplish this in some species? Poison dart frogs, they lay their eggs on land or in bromeliads. When the larvae hatch out, they're in larval form. and the parents either have to get them to water, or occasionally they have to move them from one water source to another water source. How do they accomplish this?
They transport their larvae on their back.
They do transport their larvae on their back. And so these frogs will either transport their larvae from a terrestrial oviposition site to a body of water, or as they move the larvae, or when they put the larvae in one bromeliad, so these species tend to have their larvae living in what are referred to as phytostomata. Phytostomata are just little water-filled depressions within plants. And so you see these in bromeliads. In bromeliads, the leaves grow up from a common center. And then what you will see is as rain falls down, the water collects in the center of this bromeliad. And in those bromeliads, there's a lot of times a lot of nutritional stuff. There's a lot of detritus that is rotting so that the water is filled with nitrogen and phosphorus and things like this. Also, you'll find that they grow quite a lot of algae in these phytostomata, and they also are oftentimes filled with insects and other organisms that feed on insects. And so phytostomata actually end up being very good places to rear frog offspring, tadpoles, if you live in an environment where there's a lot of rainfall, but not necessarily a lot of standing water in ponds and things like that. But what can happen to these phytostomata is they can either be depleted in terms of food supply by the larvae that live in them, or they can also dry out periodically. And so they'll have to move their larvae from one bromeliad to another bromeliad. And they'll do this by, in many cases, it's the male that does parental care in dendrobatids. The male will stick his back end down into the water in the phytostomata. The tadpoles will squirm their way up onto the back. They will suck themselves down to the back. And then after he has gathered a load of his offspring, he will hop them to the next bromeliad, stick his tail in that bromeliad. They will all slide off into that bromeliad. And he will move them as many times as he needs to as they go through development. And then we have this situation where we have actual flaps of skin that make a pouch, and the eggs are stored inside of that pouch as they develop. And then this is one of the pipids where you can see that there's this mat of skin that has grown up on the back of the pipid, and you can see these eggs that are embedded inside of this protective skin. And then, of course, this is a picture of Rheobatrachus, the gastric brooding frog, froglet emerging from the mouth of the mother who has been incubating them inside. So just lots of crazy ways of getting through these early developmental stages of being a frog or a toad. And I think we'll leave it at that. We want to talk about seasonality and the influences on seasonality on Friday. And then we'll probably finish amphibian reproduction and move into reptile reproduction, which is in chapter 9. So if you've not started on chapter 9 yet, do that by Friday. Questions? All right, I will see you tomorrow at 10:45 in White Science 219. Rose, stay on for just a minute. I just want to coordinate with you about tomorrow.